Q is focused in the central nervous system, and we are working with cells that are called glial progenitor cells. These are cells that form the cells that support the neurons. In the central nervous system, 10% of the cells are neurons. The other 90% are glia, which are required for the neurons to function properly. Our premise is that it is much easier to restore function to neurons before they die than to try to replace them and get all the proper connections and all the other difficulties of growing neurons. And that is, in a nutshell, what these cells do, is they provide normal support functions that enable neurons to remain healthy. These cells will form two types of cells: oligodendrocytes, which are the cells that form the myelin sheath that is necessary for proper signal conduction. Just like an electrical circuit, if you take the conduction off the wire doesn't conduct properly, even though it's still intact. That myelin sheath is part of a living cell, which is why no one's been able to come up with small molecules that can do that. So that is one aspect. The other thing that the glial progenitor cells, the Q cells, do is they form another type of support cell called an astrocyte. These astrocytes produce a whole range of growth factors, a lot of other trophic support, as well as directly interact with neurons, probably in mechanisms that we don't fully understand. And between these two cell types, you really have the full basis of what the neurons need to do their function. What we do is we isolate these cells at the glial progenitor stage, so we don't have to do any complex in vitro manipulation. We know the cells are at the stage that they need to be. When you implant them into the CNS of an animal, they follow the natural cues. In fact, that's where the name Q came from. They follow the natural cues, and they will form both the oligodendrocytes and the astrocytes in response to the local environment. One of the things that I think is very relevant to say is neurodegenerative diseases are very complex. It's usually not just one thing that's gone wrong that causes, the disease. When you try to treat a neurodegenerative disease with a single factor, for example, a single growth factor, that might have some benefit, but it may not cure everything that's wrong in that situation. Using the Q cells that, again, naturally form these two cell types, you have akin to a mini factory that is doing everything that occurs in a normal, healthy person that can support these neurons. So initially, we're looking at the demyelinating diseases. These would be diseases such as multiple sclerosis, transverse myelitis, potentially cerebral palsy. And then we're looking at other neurodegenerative diseases that don't involve demyelination. Our lead target there is ALS, Lou Gehrig's disease, but we also believe there are other applications for diseases such as Parkinson's and potentially even Alzheimer's. And then there are conditions, traumatic spinal cord injury, in which we believe both cell types and both mechanisms are going to be important. From the safety perspective, a significant advantage that these cells have versus systemic therapies is that it is not a systemic therapy. These cells are localized. They remain in the brain or the spinal cord. And so you do not have the issues of unknown systemic pharmacokinetics that could cause unknown systemic toxicities in going from animals to humans. But obviously, we will need to confirm that they're safe in humans. From an efficacy point, again, we're looking at disease targets where there is a fairly well-known history of the disease. So, for example, with ALS, patients are progressing very rapidly, and they're declining in multiple functions. So we will be monitoring different functions. Respiratory function is one, again, where there is a very known decline, and each patient is sort of their own control. So even in an initial phase 1/2A study, it is possible to look for measures of efficacy in addition to the measures of safety. In the multiple sclerosis area, again, there are objective measures that you can look for to see if you have improved signal conduction along a neuronal axon. And you can measure these with specific measures looking at evoked potentials. So even prior to a full phase 3 efficacy study, you may be able to see monitors that would point to efficacy in addition to safety. So we manufacture the cells by isolating them from tissues at the glial progenitor stage. We have developed a procedure that allows us to expand those cells in vitro to get a large number of cells, which we then freeze down in aliquots, which will be used in the clinical study. One of the unique properties of these cells is that when they are injected into the central nervous system, they will expand to a certain extent on their own, and they will migrate. This is a huge advantage because we don't need to inject, hundreds of millions of cells. We're projecting on the order of several million cells. We, again, don't know the exact dose until we complete the dose ranging in the animals. But the cells will expand in vivo, will migrate, and then they will terminally differentiate. Over time, the expansion falls off exponentially, and we have never seen any abnormal cell proliferation or tumor formation. The glial cells that we're developing are complementary to neurons and neuronal axons. Other companies that are working at cells that might actually expand the axons or replace the neuronal cells, I believe are much longer term. And in fact, some of these companies now are projecting that the benefits they may be seeing in animal models are actually due to growth factors produced by the cells that then act on endogenous cells. We do believe that the cells that we're providing, which provide the support for the neurons, including growth factors as well as other factors that we haven't even identified but are part of what occurs in nature, will interact appropriately with the neuronal cells. If there were another therapy that had an actual neuronal cell replacement, I think it would benefit from being used in conjunction with the glial cells.